This lecture is on the symbiotic association between Vibrio fischeri and its squid animal host, Euprimnus scolipes. I like to get into this because it's one of the foci of my research, but it's also a really great way to look at how bacterial physiology and animal physiology work together in order to establish, in this case, a beneficial relationship. And it's something you really ought to be thinking about as you get into the microbial physiology, how microbial physiology influences the animal physiology in both pathogenic and beneficial ways. And so this is a great model system. It's very easily manipulated in the lab, as I'll show you, and therefore we can do a lot of really powerful work with this association. So Vibrio fischeri is a gram-negative bacterium. It's a curved rod, thus the genus name Vibrio. It's motile. It has a tuft of polar flagella, and it makes light, so it has really interesting physiology. The bacteria will colonize a light-emitting organ. We call the light organ. So here's an adult squid. They're about the size of a walnut. This is a ventral dissection of the animal, showing you the paired light organ structures. And so here would be the gills and so forth. This is the mantle cavity, the eyes, the arms and tentacles would be here. And so inside this structure, you can see a dense population of Vibrio fischeri cells in tight conjunction with one another and intimately associated with the light organ epithelial cells. And you can see the microvilli here as well. So it gives this situation a real interesting comparison to the gut tract, for example. So it gives us the opportunity to study microbes that are doing the same kinds of things that gut microbes might do. Again, interacting intimately with microvilli in an epithelial surface. The bacteria swim into this light organ, get into this tight packed group. The cells of the light organ feed these microbes, and in exchange, the happily fed microbes, they lose their flagella, become sedentary, and then make light from this organ, so that the bacteria-derived light can be used by the squid to hunt or whatever it needs to do in its nocturnal activities. The process of luminescence is regulated by quorum sensing, which is a cell density-dependent gene expression. And so it works pretty in a simple concept. The idea is that if there's very few of you, you produce these small molecules called autoinducer that take a census, essentially, that kind of figure out how many of your siblings are around. And if these autoinducers don't detect enough siblings, nothing happens. No gene expression gets activated, or very little gene expression gets activated. These little autoinducer molecules can freely diffuse into the neighboring cells and back into the cells that produce them in order to activate gene expression. So in that way, they're taking a cell density census. So there's a low concentration of autoinducer because there's few cells, so therefore there's no gene expression. But when these cells find themselves in a higher concentration, and again, you can imagine in a natural circumstance like this, that's a high density population, the cells and their autoinducer will build up correspondingly, and eventually you'll get a critical threshold amount of these autoinducers, and gene expression will be activated. In this particular case, to form light, basically visible light. But you can also imagine this being in a pathogenic situation where you have a bacterium that invades the lungs, and there's very few of them, so they don't want to give away their presence right away because the immune system will easily swamp them out if they do. So they secretly grow, they quietly grow, and build up their autoinducers, and then when they're ready, they can overwhelm the lung with a vast amount of toxins and destroy the tissue and do their evil deeds. So in this particular case, you can look at it from an ecological perspective. The symbiosis benefits both Vibrio fischeri, as I showed you, because the bacterium gets fed, but it also benefits the squid. And so at night, the squid can go out and use their ventrally projected light, bacterial light, to help them hunt or attract mates or whatever it is that they do. We honestly really don't know. We haven't really seen this very well in nature. But we know that they do it because we can put them into sensitive equipment and see the light using the sensitive equipment. However, when this squid, who's got very big eyes, perceives the sunrise in the morning, will shoot down to the sandy bottom, expel most of its microbes, not all of them, but expel most of the microbes, leave a few behind so that they can regrow during the daytime when they're sleeping, and then at night they pop out again from the sediment, and they've got a fresh log phase population of Vibrio fischeri cells making new light. After these log phase cells have grown all night and basically become spent, the squid will see the sunrise again, head for the sediment, hide, spit out most of the bacteria, and do it again. The setup that we have is pretty straightforward. You know, we can raise animals in flowing seawater tanks. These are PVC half pipes, and here's a clutch of a few hundred eggs, and you can collect these babies and then raise the little baby squid. This is a representation of a newly hatched juvenile, and so again you can see all the features, the eyes and the mantle. Here's the light organ, and this guy's really tiny. I mean, it's about the size of a sesame seed. The squid are small and easy to handle. They hatch without bacteria, so aposymbiotic means without bacteria, and that's important because we can use these to colonize. Or we can use these squid as a vessel for our colonization assays. We can add certain kinds of bacteria to the solution, to the seawater, and see how well they colonize. The important thing here, only Vibrio fischeri colonizes the light organ. And that opens up a really interesting question. What's so special about this that all the other thousands of different kinds of microbes in seawater can't get in? Just Vibrio fischeri can get in, which is a really interesting question. It's something that we've been adding to over the decades of studying this. It takes about 10 to the third CFU per mil. That's about the infectious dose in order to get all your squid infected. And colonization correlates with luminescence. So, in the virulence world, you could dose animals with a pathogen and then take their temperature, and then fever correlates with infection. Whereas here with us, colonization correlates with luminescence. So we can then take our little squid, wash them, and then put them into fresh seawater, and then put them into a sensitive luminometer, and use luminescence as a proxy for whether or not they're colonized. The cool thing too is that the bacteria trigger developmental changes in the light organ, as I'll show you. And so that's that thing I was alluding to before about how bacterial physiology influences host physiology and ultrastructure, and vice versa. Again, the host animal physiology and ultrastructure also influences bacterial physiology and ultrastructure. And I gave you an example of that already when you look at these bacteria completely lost their flagella, for example. So here's a simplified model of the light organ colonization assay. The squid are in these tiny little vials. This is a small scintillation vial, and this is about 5 milliliters of seawater, and you have a little squid in there, and you can infect the squid with a bacterium, Vibrio fischeri or other bacteria, or mutant strains of Vibrio fischeri, just to understand what certain genes are doing in the colonization process. You let it go for a period of time, and then you measure light. Then you can crush these guys up and plate them to get an idea of how many bacteria you can recover. And you can do other fun things like compete them. So you can infect squid with a mutant and the normal wild type strain together in the same seawater, let them colonize for a while or infect for a while, and then plate them out. And then I can count the ratio of mutant to wild type in this mixture. And one of the ways we do that is we can tag the mutants with an antibiotic, for example. And so we can get kind of competitive index type assay data like this, where each of these squid or each of these circles is a squid, and they represent the ratio of wild type to mutant in each animal. And so again, this is so if any of these spheres falls on this line here, that means it's a 1-to-1 ratio. It means that it's a neutral mutation. For example, the having that mutation didn't influence the ability of the microbes to colonize the squid in equal proportions to the wild type. And then anybody over here means that the mutant, for example, this is an aminopeptidase mutant, the mutant dominated. And then any spheres to the left suggest that the wild type dominated. So again, these are ratios. And again, this is a 1-to-1 ratio. And then if you move this way, it means that the wild type predominated in this particular ratio. And so at 12 hours, you can do a time course, for example, and see how so the loss of this particular gene might have been more important early on in the colonization, but as the time wears on, it smooths out a little bit and fluctuates around the 1-to-1 ratio. So again, you can do kind of timing type assays and so forth to get a sense for the role of particular genes in colonization. This is a electron micrograph of the internal structure of the animal. And so here's the digestive tract right here, and then this is the light organ sitting on the digestive tract, almost like a saddle on a horse. And so it's a paired structure, so you have these two entryways into the light organ itself. So the bacteria would literally swim into either of these two entryways, as indicated in this really cool slide. And so this was prepared by a friend of mine, Spencer Nyholm. And so this is the mantle cavity of the squid. Here's eye and the other eye. This is its siphon. So water would flow in, and then the squid would shoot the water out, and basically as a jet propulsion system. And so the water, as it's going across the gills over here, would shoot across the light organ and then shoot out the siphon so that the squid could propel itself. And so these little structures here look like this closer up, and you can actually see the pores that the bacteria would swim into. And as they're being pushed across these, they would go into these pores and then swim deep in and colonize the light organ. And so what the data show here is, is it just a question of here's a bacterium being sucked in this turbulent wave of water and then being shot across is it just a question of the bacteria as it's shooting past jumping into the hole and accidentally some of them getting shot back out, but some of them jumping into the hole? Or is it something else? And the answer is it's something else. So what my colleague did was labeled the bacteria with a green tag and then dosed them in the seawater and asked where do they end up. And then he dissected the squid and found out that they end up as these little piles right here. So what he showed is that the squid is not just expecting the bacteria to jump in as they're being shot past the hole, but there's a mucus net that this squid produces to catch the bacteria, so that once they're caught, they can on their own time release from the mucus net and swim in, rather than hoping they can jump in as they're being shot past the holes. And that's what's pictured here. You can see these little conglomerates of cells, and then here's a close-up one showing that they're clearly a conglomerate of cells that are balled together in a glob of mucus that the squid produces. And so again, here's the representation. So you have a mixture of non-flagellated, which are not good at colonizing, and flagellated, and then random gram-negatives and gram-positives in a mixture. And so there's different layers to this, where eventually the selection process whittles it down to just the flagellated vibrio fischeri cells getting into the light organ and colonizing. And so, and this is an actual blown up image of one of the pores, showing you how these cells can be collected at the pore surface in a big mucus glob. And so again, I won't get into what these data represent, but it's just a really neat picture. The other aspect of this that I wanted you to see was the influence of Vibrio fischeri on light organ development. And so at hatching, the light organ structure looks like this. That's aposymbiotic, meaning this squid was born in seawater that doesn't have Vibrio fischeri. And then my colleagues processed it for electron microscopy. However, after a couple days, so then the seawater gets dosed with the Vibrio fischeri cells, and then you wait a couple days, the light organ looks like this. So this is gone. All this structure here is gone, and now it's just like a smooth bowling ball, and there's the little pores right there. So something that the bacteria do when they swim into these pores and conglomerate inside the light organ here, they produce signals which cause this to regress and go away. And so it's a really interesting phenomenon, this concept that the squid has this specialized organ exclusively for accepting the colonization of bacteria. And then the bacteria can modify the structure so that it suits their own needs. Once they get in, they don't want anybody else to come in. And so they get rid of this colonizing structure here. So one thing that happens is the daily expulsion of the light organ crypt contents, as I showed you already. So when this squid perceives light as it's swimming around in the water column, that triggers these organs to squeeze, essentially like squeezing a tube of toothpaste, and out comes this paste full of bacteria and other waste products. And that's how they get rid of it. That's how they get rid of the excess microbes. They'll always leave a few behind to repopulate the organ, as the squid is sleeping. And so here we're looking a little bit closer at this concept of quorum sensing and how they use it to make light. And so in a low density environment, the bacteria don't need to make light. And so they have these transmembrane sensors that, and this is Lux P and Q, and this is another sensor called AINR, and they are phosphorylated at low cell density. And so they'll transfer their phosphate groups to LuxU, and then that transfers that to another regulator called LuxO. And so LuxO phosphate activates these single strands of RNA called QRR. These single strands of RNA can block messenger RNA transcripts of this transcription factor called LIGHT-R. And that in turn blocks luminescence, because LIGHT-R is an activator of luminescence. And so under high cell density, where all these autoinducers get made, as I showed you before, these autoinducers get produced and they bind to the receptors, AINR and Lux P and Q. So there's two different kinds of autoinducer being made here. And so when that autoinducer is bound to the sensor here and the sensor here, that phosphorylation event is blocked. And as a result, LuxO never gets phosphorylated, QRR never gets made, and LIGHT-R does get made. So this messenger RNA does get made. It translates into LIGHT-R protein. LIGHT-R protein in turn activates LuxR, and then LuxR protein binds to autoinducer and activates the luminescence genes. So one of the ways we can study this is by looking at studying what genes are important in the colonization process is by sophisticated genetic tools like microarrays. And so in this in this particular example, we grew wild type and LIGHT-R mutant. So again, a mutant that's deficient in this gene, we grew these guys up in separate flasks. And then over time, we pulled samples out for RNA extraction. And then we quantified the different RNAs to get a sense for what genes are being expressed. And so again, I'm not going to get into microarrays right now, but I will in a subsequent lecture. And so the wild type, which has an active LIGHT-R, produces 9.3 units. Don't worry about the units, but 9.3 units of LIGHT-R. The mutant, which is deficient in LIGHT-R, produces virtually no LIGHT-R. I mean, that's just background, so it technically produces no LIGHT-R. This is a control, just to show you that the mutant was properly made. But what you're looking at here is an example of how we would study this type of situation, looking at capsular polysaccharide genes. And so the hypothesis is, well, just like pathogens that use their capsules to get past the immune response, maybe these beneficial symbionts are also using capsules to get past the immune response. And so in this particular case, we could ask that question and then support our hypothesis depending on what we see in the microarrays. And so here's a capsular polysaccharide operon with a bunch of genes. And then you look here at the differences in gene expression between, like, certain capsular polysaccharide biosynthesis proteins are well expressed in the wild type, but barely expressed in the mutant, suggesting that you take LIDAR away and expression of this particular capsular polysaccharide goes away. And so that supports the hypothesis then that LIDAR has something to do with capsules, and in turn might have something to do with the ability of the bacteria to colonize. So we already know that LIDAR has a lot to do with luminescence production, and that was something that we verified through experimentation and something you guys will work on in the lab. But what about other functions? And again, this is an example of that here, where these capsular polysaccharide genes are definitely downregulated when this transcription factor is removed, suggesting that it enhances their expression when it's around. And so one of the examples of that is, in support of that, one of the examples of experiments in support of that is this, just looking at the colonies. Here's a colony of the mutant PMF8, here's a colony of the wild type. And you can see there's a significant difference in opacity. This guy looks pretty opaque, suggesting that it's making more polysaccharide, capsular polysaccharide slime, whereas this guy is not. And that, again, supports this. So, and I'll skip past some of these, but I really want you to see that, that's an example this is just an example of how we look at this from how a modern microbial physiology lab looks at a big problem like this, like the influence of microbial physiology on the animal host. And here's another example of that, how we discover genes required for squid colonization. This is the Mandel paper, and I've supplied this to you so you can read through it if you're interested. But in this particular case, what we do is we make a whole bunch of random mutants, and then we, in that pool of mutants, they're all labeled, and that's why these are different colored. So each of the mutants is labeled differently, and then we put that into the squid. And then we can do that in culture medium. We can put a population of these pile of mutants into the culture medium and then see what comes out, like who wins in the culture medium. And then we can put these random mutants into the squid and we can see who comes out of the squid. And the idea here is, if we find something that, if we find a mutant that grows just fine here in culture, but is completely depleted when we try to recover it from squid, we can look at that more closely and ask ourselves, is that a really important colonization factor? Because it's not represented here. Now, if there's a bacterium, a mutant that's not represented here and not represented here, we don't care about it as much because it means that, something vital, a growth factor was depleted and so the microbe can't grow. So we don't care because that doesn't surprise us that if a microbe is deficient in a significant growth factor, it wouldn't be able to grow in the squid. But what we want are evolutionary adaptations for growing here, not evolutionary adaptations for growing in general. And so again, using this approach that's outlined in the Mandel paper, you can get a sense for how we use high throughput, large-scale mutagenesis libraries in order to understand what genes are important in this really sophisticated symbiotic association.